When researchers at the Beijing Genomics Institute published their findings in 2018, the scientific community went silent. Music. They had just sequenced the genomes of over 140,000 Chinese individuals and compared them to European genetic databases. What they found was not just unexpected, it completely rewrote our understanding of human evolution, musical migration, and what makes populations different from each other at the genetic level. The results were so controversial that 3 peer reviewers initially rejected the paper, not because the science was bad, but because the implications were that profound. Here is what they discovered. Before we dive into what shocked everyone, you need to understand what scientists expected to find. For decades, the mainstream narrative about human ancestry followed a simple path. Modern humans evolved in Africa around 300,000 years ago. Then, roughly 70,000 years ago, small groups migrated out of Africa and spread across the world. Europeans went one direction, Asians went another. Over thousands of years, these isolated populations developed distinct physical features: skin color, eye shape, hair texture. But underneath those surface differences, scientists believed we were all basically the same at the genetic level. That assumption held strong for years. Sure, we knew about minor variations, lactose tolerance in Europeans, altitude adaptation in Tibetans, but these were seen as tiny tweaks, nothing fundamental. The genetic blueprint was assumed to be nearly identical across all human populations. Then, the Beijing Genomics Institute started their project. Music. They were not looking for controversy. They were simply trying to map genetic variations across China's massive population. China has 1.4 billion people spread across vast geographic regions. The goal was practical: better healthcare, more accurate disease prediction, and personalized medicine tailored to Chinese genetics. But when they started comparing Chinese genomes to European databases, the numbers did not make sense. The first shock came from something called genetic diversity. Think of genetic diversity as the variety of genetic differences within a population. More diversity means more genetic variation. Less diversity means people are more genetically similar to each other. Scientists expected Asian populations to have lower genetic diversity than Africans, since Asians descended from small founder groups that left Africa. That part was correct. But what stunned researchers was how different Chinese genetic diversity looked compared to Europeans. European populations showed relatively uniform genetic diversity across the continent. Whether you sampled DNA from someone in Sweden or someone in Greece, the variation levels were similar, but Chinese populations varied wildly depending on the region. Southern Chinese populations showed genetic variation patterns that were completely distinct from northern Chinese populations. These were not small differences. In some measures, the genetic distance between a Han Chinese person from Beijing and a Han Chinese person from Guangdong was greater than the genetic distance between a Swedish person and a Greek person. That discovery alone was surprising, but it was just the beginning. The second finding hit even harder. When researchers analyzed specific gene variants, they found thousands of genetic mutations, that were either extremely common in Chinese populations but rare in Europeans, or the other way around. We are not talking about cosmetic differences like eye color genes. These were genes affecting metabolism, immune function, brain development, and disease susceptibility. One example stood out immediately: the gene variant EDARV370A. This mutation affects the development of hair, teeth, and sweat glands. In Chinese populations, this variant appears in over 90% of individuals. In Europeans, less than 5 route. That's not a minor variation. That's a fundamental genetic difference, present in nearly every Chinese person, but almost no Europeans. But here's where it gets controversial. The EDAR gene does not just affect hair texture. It also influences the density of mammary glands and the branching patterns of neural connections during fetal development. Some researchers began asking uncomfortable questions. If this gene affects brain structure during development, and it is present in 90% of Chinese people but only become a scene of Europeans, what does that mean? The paper did not go there, but the question hung in the air. Then came the third discovery, and this one shattered assumptions completely. Scientists found evidence of selective sweeps. A selective sweep happens when a genetic mutation becomes common in a population very quickly because it provides a major survival advantage. It is evolution in fast-forward mode. The mutation can spread through a population in just a few thousand years instead of hundreds of thousands. European populations showed evidence of selective sweeps for genes related to skin pigmentation and lactose tolerance. That made sense. Lighter skin helps with vitamin D production in northern climates. Lactose tolerance helps when dairy becomes a major food source. These were well-known adaptations, but Chinese populations showed selective sweeps in completely different categories. Genes related to fat metabolism, genes affecting neurotransmitter regulation, genes influencing spatial reasoning and memory formation. Here is the kicker. These selective sweeps happened recently, within the last 10,000 to 30,000 years. That timeline matters. 10,000 to 30,000 years ago is after the migration out of Africa. These genetic changes were not inherited from a common ancestor. They evolved independently in Chinese populations after the split from European lineages. The implications were staggering. It meant that Chinese and European populations had not just drifted apart through random genetic mutations. They had been shaped by different evolutionary pressures, selecting for different traits, optimizing for different environments and survival challenges. And those differences went deeper than anyone expected. The fourth finding pushed things even further. Researchers discovered what they called ghost DNA. This is genetic material that does not match any known human lineage. It is not from Homo sapiens who migrated out of Africa. It is not from Neanderthals who interbred with Europeans. It is not from Denisovans who interbred with some Asian populations. Music. It is something else, something we have not identified yet. European populations carry about 2% Neanderthal DNA on average. Some Asian populations carry Denisovan DNA, but the Beijing study found segments of Chinese genomes music that did not match any of these known archaic human species. The percentages were small, less than 1%, but they were there, consistent, reproducible. Where did this mystery DNA come from? One hypothesis is that there was another archaic human species in Asia that we have not discovered yet. Modern Chinese populations interbred with this unknown group, just like Europeans interbred with Neanderthals. The genetic signature remains, but we have not found the fossils yet. Another hypothesis is music, even stranger. Some researchers suggest this DNA might not be from interbreeding at all. It could be from convergent evolution, where similar genetic mutations arise independently in different populations because they solve the same environmental challenges. But that explanation struggles to account for the specific pattern, music, seen in the data. Either way, Chinese populations carry genetic material that Europeans do not, and we do not fully understand what it is or where it came from. Now, here is where the controversy exploded. Remember, all of this research was done for medical purposes. The goal was to improve healthcare by understanding genetic differences that affect disease risk. That is where the findings became impossible to ignore. Chinese populations showed significantly different genetic risk profiles for almost every major disease category compared to Europeans. Cardiovascular disease, diabetes, Alzheimer's, cancer, autoimmune disorders. The genetic variants that increase risk in European populations often had completely different effects in Chinese populations, or were not present at all. Take type 2 diabetes. In European populations, the strongest genetic risk factors are variants in genes like TCF7L2 and PPRG. Music. These variants dramatically increase diabetes risk in Europeans, but in Chinese populations, those same variants showed much weaker associations. Instead, Chinese diabetes risk was driven by completely different gene variants that barely mattered in European populations. This has massive implications for medicine. For decades, most drug trials and medical research have been conducted on European populations. The drugs that work for Europeans are assumed to work for everyone. If the genetic architecture of disease is fundamentally different between populations, those drugs might not work the same way or at all. That is exactly what researchers started finding. Certain medications used to treat high blood pressure worked well in European patients, but showed reduced effectiveness in Chinese patients. Chemotherapy drugs, optimized for European genetics, had different response rates in Chinese populations. Even pain medications were metabolized differently. The medical community was forced to confront an uncomfortable truth. Genetics matter more than we admitted. Population-level genetic differences are real, significant, and clinically relevant. Publishing these findings opened a Pandora's box. Once you acknowledge that populations differ genetically in ways that affect health, disease, and drug response, the question becomes, what else might be different? The paper carefully avoided discussing cognitive traits, behavioral tendencies, or anything that could be interpreted as ranking populations by genetic quality. The researchers knew the political minefield they were walking into, but the data was there. Gene variants affecting neurotransmitter function, variants affecting brain structure development, variants linked to memory, learning, and spatial reasoning in animal models. 3 peer reviewers rejected the paper initially. They did not reject it because the methodology was flawed. They rejected it because they worried about how the findings might be misused. One reviewer warned that publishing this data could fuel racist narratives. Another argued that the social harm outweighed the scientific benefit. The research team pushed back. They argued that suppressing scientifically accurate data for political reasons was itself unethical. If genetic differences exist and affect medical outcomes, doctors need to know. Ignoring reality does not make it go away. It just leads to worse healthcare. The paper was eventually published, but with heavy caveats. The authors emphasized that genetic differences between populations are small compared to genetic variation within populations. They stressed that these findings say nothing about individual capabilities or worth. They warned against drawing conclusions beyond the medical data. Once the paper was public, the debate exploded. Some researchers embraced the findings and called for more population-specific medical research. Others accused the authors of scientific racism. The controversy became so intense that several researchers involved in the study stopped giving interviews. Meanwhile, other teams began replicating the findings. A study from Japan found similar patterns when comparing Japanese genomes to European databases. A South Korean team published comparable results. The data kept pointing in the same direction. Asian and European populations are more genetically distinct than previous models suggested. These distinctions have real-world consequences that go far beyond medicine. One of the most fascinating follow-up discoveries came from researchers studying altitude adaptation. Tibetan populations can survive at elevations that would cause severe altitude sickness in most people. Scientists assumed this was due to Denisovan DNA. Tibetans do carry Denisovan genetic material, including a variant of the EPS1 gene that helps regulate oxygen levels in the blood. When researchers looked closer, they found that Han Chinese populations living at sea level also carried different variants in oxygen metabolism genes compared to Europeans. Those variants did not provide the extreme altitude tolerance seen in Tibetans, but they did seem to optimize oxygen usage in different ways. The genetic differences were not just about adapting to extreme environments. They were about fine-tuning metabolism for the specific environmental conditions populations faced over thousands of years. Another research team focused on diet-related genes. They found that Chinese populations showed genetic adaptations to high-starch diets that were distinct from European adaptations to dairy-heavy diets. The AMY1 gene, which produces an enzyme that breaks down starches, showed significantly more copy number variations in Chinese populations. More copies of the gene meant more enzyme production and more efficient starch digestion. This makes perfect sense when you consider agricultural history. Rice cultivation in China dates back over 10,000 years. Music. A high-starch diet would create evolutionary pressure favoring individuals who could efficiently digest and metabolize those starches. Europeans, meanwhile, developed lactase persistence because dairy became a crucial food source in northern climates, where agriculture was harder. These dietary adaptations cascade into other biological systems. Starch metabolism affects insulin response, which affects fat storage, which in turn affects cardiovascular risk. The genetic adaptations that helped ancient Chinese populations thrive on rice-based diets created different metabolic baselines that persist today. Then, there is the microbiome connection. Gut bacteria are influenced by both diet and genetics. Chinese populations harbor different gut bacterial compositions compared to European populations, and those differences are partly driven by the genetic variants affecting digestion and metabolism. This creates a feedback loop. Genes shape the microbiome. The microbiome shapes health outcomes. The whole system is population-specific, but perhaps the most profound implication comes from neuroscience research. This is where things get really uncomfortable, so most researchers avoid discussing it publicly. But the data exists. Several gene variants that differ significantly between Chinese and European populations are known to affect brain structure and function.
Anomaly detected. Levels are critical.
The ASPM
This can't be right.
And
Rerun the simulation.
Genes,
the energy
which are
signature is
involved
off
in
the charts.
brain size regulation and neuronal development, show different variant frequencies between populations. The DRD4 gene, which affects dopamine signaling and is linked, music, to novelty-seeking behavior, shows population-specific patterns. Does this mean anything meaningful about cognition or behavior? Honestly, we don't know. The science of linking specific genes to complex traits like intelligence or personality is still in its infancy. Most cognitive abilities are influenced by thousands of genes working together, plus environmental factors, plus cultural influences. Trying to draw conclusions from a handful of gene variants is scientifically premature. But here's what we do know. Brain development is influenced by genetics. Neurotransmitter systems are influenced by genetics. And if populations differ significantly in genes that affect these systems, there is at least a theoretical possibility that population-level cognitive or behavioral patterns could have genetic components. Most researchers refuse to touch music sophisticated enough to answer these questions properly. But the Beijing Genomics Institute study opened a door that cannot be closed. We now know that Chinese and European populations are more genetically distinct than we previously believed. We know those differences affect disease risk, drug response, music, and metabolic function. And we know that some of those differences involve genes that affect brain development and neurotransmitter function. What we do with that knowledge is the real question. The optimistic view says this research will revolutionize personalized medicine. Imagine a future where your doctor prescribes medications based on your specific genetic ancestry, maximizing effectiveness and minimizing side effects. Imagine cancer treatments tailored to the genetic mutations most common in your population. Imagine disease prevention strategies that account for your population-specific risk factors. That future is already beginning. Several pharmaceutical companies now conduct separate drug trials in Asian populations, specifically because of findings like these. Genetic testing companies are developing ancestry-specific health risk assessments. Medical schools are starting to teach population genetics as part of standard curriculum. But there is a darker possibility. If genetic differences between populations...
This acceleration rate is unprecedented.
Will that fuel discrimination?
We need to verify these figures immediately.
Will it be used to justify unequal treatment or reinforce harmful stereotypes? History shows that scientific findings about human differences have been weaponized before. Phrenology, eugenics, scientific racism. All of these movements claim to be based on objective science. The difference now is that the science is actually rigorous. The genetic differences are real and measurable. But real does not mean what people think it means. Real does not mean superior or inferior. Real does not mean destiny. Real just means there is biological variation between populations that evolved in different environments over thousands of years. And here is the crucial point that gets lost in these debates. The genetic differences between individuals within a population are much larger than the average differences between populations. If you pick two random Chinese people, they will differ from each other genetically far more than the average Chinese person differs from the average European person. What the Beijing study showed is that the pattern of variation is different between populations. The specific genes that vary reflect different evolutionary histories. But it does not create a hierarchy. It creates diversity. Diversity that has medical implications we are only beginning to understand. When the research team published their findings, they included a statement worth reading in full. They wrote that their results demonstrate that human populations have experienced different selective pressures and carry different genetic architectures for complex traits. This diversity should be celebrated as a source of human resilience and adaptability, not used to rank populations or justify discrimination. The goal of this research is to improve health outcomes for all people by understanding how genetic variation affects disease risk and treatment response in different populations. That statement tries to thread an impossible needle, acknowledge real differences while insisting those differences do not imply superiority, use the findings for good while preventing misuse. It is a noble attempt. Whether it is achievable is another question. Since the Beijing study was published, the field has moved forward carefully. More research is being done on population-specific genetics, always with extensive ethical oversight. The focus remains firmly on medical applications, disease risk, drug response, personalized healthcare. But the fundamental questions remain unanswered. If populations differ genetically in ways that affect disease susceptibility and drug metabolism, what else might be different? Some of the genes that differ between Chinese and European populations relate to brain development, behavioral regulation. What do those differences actually mean? Scientists are working on answering these questions, but progress is slow. It is not just a matter of collecting more data. It is about developing better tools to understand how thousands of genes interact to produce complex traits. It is about separating genetic influences from environmental and cultural factors. It is about asking questions that society may not be ready to hear answers to. And that brings us back to where we started. When the Beijing Genomics Institute compared Chinese DNA to European DNA, they expected to find minor variations, tweaks in a fundamentally identical blueprint. What they found instead was evidence of distinct evolutionary trajectories, different selective pressures, and genetic differences that affect health, metabolism, and potentially much more. The results shocked everyone because they challenged a comforting narrative, the idea that we are all the same under the skin, that human populations are interchangeable genetic units that just happen to look different on the surface. The Beijing study showed that is not quite true. We are more different than we thought, but also more interesting, more complex, more adapted to our specific histories and environments. What we do with that knowledge will define the next chapter of genetic research. Use it to improve medicine and reduce suffering. Music, that is the hope. Use it to divide and discriminate, that is the fear. Musik, the science itself is neutral. The data just is, but the interpretation, the application, the way these findings shape society is up to us. The results shocked everyone. Now we have to decide what comes next.